Now we go to the clinical approach to drug delivery. So this is the clinical phase. Before this, we were discussing about preclinical phase, and now this is clinical phase. So clinical phase involves clinical trials. There are 3 or 4 steps of clinical trials. Phase 1 trial usually involves healthy volunteers to determine safety and dosing. So there are no diseased individuals or subjects are used in the phase 1 trial. And the phase 2 trials are used to get an initial reading of efficacy and further explore safety in small numbers of patients having the disease targeted by the NCE. And phase 3 trials are large pivotal trials to determine safety and efficacy in sufficiently large numbers of patients with the targeted disease. If safety and efficacy are adequately proved, clinical testing may stop at this step and the NCE advances to the new drug application stage. Okay, so phase 2 and phase 3 are quite similar. The main difference between the two phases are just the number of participants. So phase 2 only involves hundreds of subjects, but phase 3 involves thousands of subjects. Phase 4 are post-approval trials. There are sometimes a condition attached by the DFA, also called post-market surveillance studies. Okay, after the drug is marketed in the market, then there will be phase 4, whereby there will be a surveillance to see whether those drugs are actually producing any side effects or not, and so on. So the process of defining characteristics of the drug does not stop once an NCE begins human clinical trials. In addition to the tests required to move a novel drug into the clinic for the first time, manufacturers must ensure that any long-term or chronic toxicities are well defined, including effects on systems not previously monitored. For example, the fertility, reproduction, immune system. So they must also test the compound for its potential to cause cancer. Okay, even though the NCE is aiming for something related to maybe for cardiovascular disease, manufacturers must also uh, observe the effect on the fertility, carcinogenicity, and so on. And if a compound emerges from these tests with an acceptable toxicity and safety profile, and the company can further show it has the desired effect in clinical trials, then the NCE portfolio of evidence can be submitted for marketing approval in the various countries where the manufacturer plans to sell it. So in the US, this process is called a new drug application or NDA, and in Malaysia, it can be uh, forwarded to NPRA. Most NCEs fail during drug development, either because they have unacceptable toxicity or because they simply do not have the intended effect on the targeted disease as shown in clinical trials. So as I mentioned earlier, I think in the first or second video, there are many reasons that can contribute to the failure of the drug uh, in clinical phases. So these are some of the examples whereby an NCE can be turned down. So the cost of developing a drug, the full cost of bringing a new drug to market from discovery through clinical trials to approval is complex and controversial. Why? Typically, companies spend tens to hundreds of millions of US dollars. One element of the complexity is that the much publicized final numbers often not only include the out-of-pocket expenses for conducting a series of phase 1 until 3 clinical trials, but also the capital cost of the long period 10 or more years during which the company must cover out-of-pocket costs for preclinical drug discovery. So sometimes the figure that is given by the pharmaceutical company is not really accurate, because they did not only count the money that they have spent on developing the drug, but they also monetize the period, the duration of years that they have spent in developing the drug. So let's say for 1 year, they estimate demonetize that 1 year equals to how many millions. Additionally, companies often do not report whether a given figure includes the capitalized cost or comprises only out-of-pocket expenses or both. Another element of complexity is that all estimates are based on confidential information controlled by drug companies, released by them voluntarily, leading to inability to verify costs. The numbers are controversial as drug companies use them to justify the prices of their drugs, and various advocates for lower drug prices have challenged them. The controversy is not only between high and low, but also the high numbers may vary considerably for the manifold factors in drug development. Because this is about business, of course, when you talk about business, you cannot publicize your financial report, your detailed financial report to the public because it's confidential. It's the secret of the company. So normally they will just give a rough number, a rough figure of the cost. And no one can check this because it's the secret of the company. And that's why you can, if you refer many other sources, you will see that the cost of developing a drug can range from millions to billions because of this reason. Candidates for a new drug to treat a disease might theoretically include from 5,000 to 10,000 chemical compounds. On average, about 250 of these show sufficient promise for further evaluation using lab tests, mice, and other test animals. Typically, about 10 of these qualify for tests on humans. A study conducted by Tufts Center for the Study of Drug Development, covering from 1980s to 1960s, found that only 21.5% of drugs that started phase 1 trials were eventually approved for marketing. Okay, only 25% from phase 1 trials were eventually marketed. In the time period of 2016 to 15, the success rate actually declines to 9.6%. The high failure rates associated with pharmaceutical development are referred to as the attrition rate problem. Careful decision making during drug development is essential to avoid costly failures. In many cases, intelligent program and clinical trial design can prevent false negative results. Well-designed dose-finding studies and comparisons against both placebo and a gold standard treatment arm play a major role in achieving reliable data. Okay, next is drug characterization. The pre-formulation studies will attempt to characterize the drug in a number of respects. Okay, the pre-formulation studies will attempt to characterize the drug in a number of respects. They are spectroscopy. The function of spectroscopy is to produce a simple method for analyzing the drug, maybe in terms of what they call mass, molecular mass, or molecular weight, and then solubility for identifying the best salt to develop and for producing liquid dosage forms, melting point, assay stability, microscopy, powder flow and compression properties, and last one is excipient compatibility. So these are some of the drug characterization that is done on the drug. Okay, last one is dosage form. What is dosage form? At some stage, a decision needs to be made about the dosage forms for the delivery of the drug. For example, a tablet, capsule, injection, syrup, emulsion, and so on. The factors that determine which dosage form is or are to be used are many and involve marketing considerations apart from scientific considerations. Okay, there are many factors that contribute to the decision on what dosage form that we should select for that particular drug. Types of dosage forms. Nowadays, there are many different dosage forms, including the 3 examples given above. and they all have their relative advantages and disadvantages. So these are some of the dosage forms, tablets and capsules. Remarks: convenient and commonest dosage forms, but likely to be no good if the drug cannot be absorbed in the alimentary tract or if the patient, for example a child, cannot swallow them. Okay, so this is the most common, the most convenient, because the patients just have to swallow and drink some water. But if the patient is unresponsive or not cooperative, just like a child or a patient that is not conscious, then it's no use. Then we have to think for other dosage forms. Injections and infusion, rapid action, but impractical for treating chronic illnesses. So for chronic illness, for chronic disease, because it's taking a long time, many years, so it's really not practical to use injection. Pessaries and suppositories can deliver the drug to local area where required but have limited general use. Solution, suspensions, and elixirs, useful for children and the elderly but are bulky and less useful if the drug is unpalatable or unstable in the presence of water. And then ointments, creams, and pain. Use is restricted to topical application. Aerosols and dry powder inhalation, good for drugs required but can be difficult to administer the dose correctly. And transdermal patches, convenient if the dose need to be released over a long period. The therapeutic considerations play an important role in deciding the dosage form to formulate. Here are a few examples on how, this is a real case scenario on how to decide on a dosage form. Okay, a tablet is not suitable dosage form if the drug cannot be absorbed in the alimentary tract, unless of course it is required to treat an ailment in the tract itself, such as a gut infection. Instead of a tablet, an injection might be suitable alternative. Okay, so if the drug cannot be absorbed in the GI tract, then it is not suitable, unless it is a drug, for example, a drug for what do you call it, for gastric. Okay, so you want the drug to be acting in the stomach itself, so that can be used as a tablet. And even if the drug can be absorbed in the alimentary tract, let's say the intestine, as the tablet will still be unsatisfactory if the drug is destroyed in the stomach acid. Okay, if the drug is not stable in the hydrochloric acid in the stomach, then it is still not suitable for tablet. In such a case, the tablet might be entry-coated to prevent drug destruction whilst the tablet is passing through the stomach. But there is a way on how to avoid this, is by coating. It's by coating the tablet so that it does not dissolve or it does not disintegrate in the acidic condition in the stomach. By the time the tablet reaches the intestine, the coating has dissolved, liberating the drug for absorption through the intestinal wall. So it can be formulated so that the coating will be gone in the stomach, and then by the time it reaches the intestine, it can disintegrate and ready to be absorbed by the intestinal wall. Drugs which need to act immediately, for example, bronchodilator drugs for treating asthmatic attacks, where the airways suddenly become so constricted that the patient has difficulty in breathing, are best delivered by inhalation directly to the lungs, where they can rapidly dilate the airways, rather than being swallowed in a tablet with a consequently delay in action whilst the drug is absorbed and delivered to the lungs. So to be effective, a drug must reach in desired concentration to the part of the body where it is required to act, and ideally must be maintained at concentration for the appropriate period of time. This goal is influenced by the key interactions which takes place between the drug and the body after the drug has been administered. These are the ADME: absorption, distribution, metabolism, elimination, that you have learned in your pharmacology. So these processes are referred to as ADME in short. The study of the pharmaceutical factors which affect the fate of the drug after administration is called biopharmaceutics. And these factors will need to be evaluated in the development of a new drug.